Hello and welcome to this video on what proteins are. Proteins are the end result of putting amino acids together. The chains of amino acids can be very short or very long, but as a rule anything shorter than 30 amino acids or residues is not considered a protein but a polypeptide. There is also the three-dimensional nature of proteins. They are not just simple linear chains, but instead have to shift and form in a way that the individual amino acids that make them can be chemically stable. Proteins begin by taking a codon or set of 3 nucleotides and reading this to match a specific amino acid. This is then added to the end of a peptide chain. As you build up the peptide chain, it then becomes a protein. Most proteins are at least somewhat unique in their structure, and this is because of the way it folds and twists. As mentioned, all the individual amino acids need to be chemically stable. A lot of the time, this is either the charges on the side of the amino acid, or the amino acid is hydrophobic, that is, it doesn't like to be exposed to water. Just about all proteins will fold because of this, and you require no energy to be put into the system. It is purely based on the gradient of chemical energy within them. Admittedly, there are some more specific and highly evolved proteins that do require more cellular processes to make them happen. Notably, they'll need some sort of chaperone molecule to ensure that they don't fold into the shape they're meant to be chemically stable. Because of the way proteins exist and the fact that they're made up of not just individual protein chains, but in fact multiple side structures, they get broken down into different levels of organization. There is your primary structure, which is basically just your amino acid sequence, that theoretically linear chain of amino acids, but realistically not so much. You then have your secondary structures. Secondary structures are how the different parts of that primary structure interact, mostly through hydrogen bonds. You'll see this in the form of alpha helix and beta sheets. This is one thing that also raises the other attribute of secondary structures, and that they are often repeating structures. That is, a series of amino acid sequences that continue several times over. Next are the tertiary structures. Tertiary structure relates more to how two parts of the same protein but are well separated interact. So for example, if you had 2 alpha helices, the internal workings of one would come under secondary structure, while the interaction between the two would be considered a tertiary structure interaction. Common examples of this are things like disulfide bonds. The disulfide bonds allow for those two very distinct parts of the protein to interact and maintain their stability with each other. Next are your quaternary structures, and this is when you get different proteins all interacting together. This is something you'd see with things like your antibodies and with the hemoglobin in your red blood cells. By having the multiple polypeptide chains or proteins interacting in this way, you get what is effectively one very large and complex protein made up of a multitude of smaller and somewhat less complex proteins. Finally, there are quinary structures, and these are less used now, primarily because they are very specific, but also generally quite transient. They often only exist for a short time frame and inside of a living cell, where they create an interaction to allow for something else to occur. Most of the time, the best example is going to be something like a cell receptor. Going back to some of the information mentioned, in a way similar to sugars, there are 2 significant things in protein you need to look out for. They are beta sheets, or pleated sheets, and alpha helices. You see something developing in sugars as well. The beta sheets are a regular recurring pattern where, due to the secondary structure, you get something looking like a corrugation in cardboard. They normally have a hydrogen bond which helps them keep that shape, but that also means that because of how they're structured from amino acids, they get twisted, and that twisting, when held together with the hydrogen bonds, leads to a hill and valley-like appearance. The alpha helices is somewhat different in that it is a helices. That is, it goes around in a circle just like your DNA. The pattern generally occurs because the amine group, which is at one end of an amino acid, has a hydrogen bond to the carboxyl group of an amino acid a short distance away. This interaction of the two allows for that helix-like shape to occur because you not only have a regular recurring pattern, but that regular recurring pattern has a consistent linkage. It's the same way that if you were to try and lay railway tracks, you have consistent railway ties at a set distance apart, and that's what keeps them straight. In theory, you could use the same idea, but allow them to create a structure that is, let's say, less straight and linear. This is the main features of your secondary structures, but tertiary structures are the next to talk about, and they will change the three-dimensional shape of the protein. If you were to look at just primary and secondary structure together, they would still remain relatively linear. You don't really get any turns or twists with it. Once you start to bring in the effects of tertiary structure, you begin to see a lot of that three-dimensionality come into play. And that's because of how you can connect, for example, alpha helices or pleated sheets together using these bonds, and that can cause up to a 180-degree turn or right back on itself, thereby allowing the protein to achieve a much more interesting and useful shape. Tertiary structures don't just need to look at a single individual amino acid. They can use the entirety of a secondary structure domain. That is, the entire region can somehow play a role in forcing this to happen. This is also where the Quaternary or fourth level of protein structure comes into play. And this is arguably, as mentioned, the most advanced level of protein with regards to a long-term stable protein. The complement cascade is an example of what happens with quaternary structures. In that instance, you see how the different antibodies come together, and they collectively form a large single unit from many smaller subunits. The arrangement of these all comes into how the individual proteins as a single entity interact with the other single entities. And so you see that there's not just the same localized influence of the secondary, tertiary structures, but that they then interact with the others. And this becomes really important when we're looking at how multiple proteins can fit together. If you didn't have the same interaction and, let's say, cooperation or ability to mesh, you couldn't ever get a stable, large protein to exist. You simply couldn't have the individual units that make up the big unit work that way. This is where things like the individual charges on amino acids and the overall total charge of a protein are really important. If you have a relatively nonpolar or non-charged protein, it can generally cooperate with anything else to an extent. At the same time, a very polar or non-polar protein creates problems, although most of the time you'll find that they are nearly always polar if they have a charge like that. This again feeds into how they can interact and whether or not they can mesh. Finally, there are those quinary structures we mentioned early on, the short-lived and relatively unstable structures. They really only occur because you get constant interactions, especially on the cell surface, where interactions between different proteins lead to much more complex structures. But again, because they're unstable or they have a very short-lived and specific purpose, they tend to disappear reasonably quickly. Proteins are much more than the sum of their parts, and they're certainly more than just the thing that makes up meat, for instance. Those parts that we are very familiar with are very complex, and the depth to them is incredibly deep. This is one reason why it was thought for a long time that protein and not DNA is what gave life instruction. We now know that DNA, through the interaction of multiple different parts and regulation pathways, can actually have even more complexity to it, if only because of the length and size of it and how it codes from that. With that in mind, protein is still very complex, and we've only scratched the surface by telling you how the multiple layers of protein complexity are roughly defined. Looking at any individual example, you will see that they are very difficult to understand, so much so that, for example, citizen science projects like Fold It exist, because it's much easier to get an individual to figure out how a protein goes together and interacts than it is to try and develop an algorithm that does the same job. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions you have below.